on training practicals are going on for this training. Yesterday, Sir has covered the topics on immunodiagnostics and the antigen preparation, purification of antigen, and immunoassay development. And today, he is going to deliver his topic on good practices in sample preparation for sequencing, as well as Sanger sequencing and troubleshooting. So all online participants are requested to please mute your mic, and if you have any doubts, queries, please put it in the chat box, or after the lecture also you can ask the questions. So now I invite and request Dr. Mangesh Bede sir to continue his topic. Sir, please.
Okay, so thank you very much, Dr. Vaidya, for today's introduction. So we are again jumping back to DNA and gene and how to sequence, because this is one of our main goal to identify the pathogen. But the second goal is not only identify based on the PCR whether there is band or not, but we need to identify at nucleotide level, and we need to many times we need to genotype it and compare with the sequences available in the gene bank and make the phylogeny, that is molecular epidemiology work. So we will first see some how DNA sequencing works and why we need DNA sequencing when there is NGS, next generation sequencing, why we need Sanger sequencing. So DNA sequencing is one of the most, is not working. Okay. DNA sequencing still remains one of the most used experimental approach in molecular biology after the SDS page. So do you know who has the maximum number of citation in the world? Is Lamelli. Lamelli, who discovered the SDS page, who has in million, and the second one is Sanger, and third one is Bradford. So if you invent any method, you will be also in millions of citations. So the Lamelli, Sanger, and Bradford, they have something common. Okay, because the Sanger sequencing was previously based on the SDS page or page, polyacryl amide gel electrophoresis. So it was not capillary based, but it was gel based. So as the Lamelli invented the separation of different molecules through polyacrylamide gel electrophoresis, the Sanger was able to separate his nucleotides through the polyacrylamide gel electrophoresis. And where is Bradford? Bradford was used as a negative control or Bradford assay to see whether there is some protein contamination in DNA before sequencing. Okay. So where we need Sanger sequencing? First, 5% variation detection with Sanger sequencing, that is variant detection, genotyping. We need to identify each and every nucleotide. And this is extremely important, that is Sanger NGS confirmation, the second point. Although you have NGS and you see some new variants or gene, you need to go back and confirm with the Sanger sequencing. So the myth that NGS is good, NGS is awesome is fine, but it is not 100% sure that all the variants you are getting, that is different genotype or change in the nucleotide, you have to go back and confirm with the Sanger. This is in case of the genomics. In case of proteomics, for example, you have MALDI-TOF, you have LC-MS-MS, you have Orbitrap, for example, all multi-million dollars instruments. And whenever I was sending the data to uh, good journal like Journal of Proteomics or uh, some basic journals, they were asking me, okay, show me data from SDS page. That cost me around 200 euros. Okay, so just compare some million euro machine, and reviewers were asking, show me data from SDS page. So that is the standard machine and standard protocol where I see my results, and I see the degradation of protein, I see the quality of protein, I see what non-specific multitope will not show you that. So in proteomics, you have SDS page, ELISA, basic. Here you have Sanger sequencing, the basic. Okay, Sanger sequencing, you cannot change Sanger sequencing, whatever machine will come. Okay, NGS has not received any Nobel Prize. This has, so Sanger has received Nobel Prize. And then we need to have some microbiome and all this. So after microbiome analysis, you have to go back and sequence on the Sanger. So Sanger sequencing still remains as a basic tool for confirmation of all nucleotide identifications. So have you seen the electrophotogram or chromatogram, we say? So this is the baddest chromatogram ever you can see. You should not have this kind of uh, results, but often when you send samples for the sequencing, you get this kind of result. So today what we are going to see is why we get this kind of results and how to avoid that. But before, we will go for the basic how it works, and then you will identify how to avoid these problems. So first time you will get this, the second time you will have this kind of reaction, the third time you have this kind of reaction, and you don't know what to do to avoid your bad results. And this is a typical feature of bad result. So whenever you will get a flat file from the company, okay, so this is n. This is a typical feature. You don't try to call back and, oh, I don't see anything, because your sample is bad. Okay, this is 101% sure that you send your sample in bad condition. So this is a typical old school Sanger sequencing. Do you know what is this? Huh? This is PCR machine. Okay, so I have seen this PCR machine actually, and I have worked on this kind of PCR machine. So in Spain, when I was working, still in museum it was, and people were using those who had that time in 2001, when they were having older people, like of 60-year-old. In Europe, they can work till 70 actually, 70 years of age, still they work here. And they were putting a denaturation. This is the second annealing and extension. So they were putting simple like this. Okay, and PCR machine was working. So the first step is PCR for any sequencing, and then they were having Sanger sequencing chemistry or DDNTP chemistry. So you were asking yesterday what is that DDNTP? You will see what is that today. And then they were putting this after this chemistry, they were putting their sample on the polyacrylamide gel. Okay, so it was really big polyacrylamide gel. Till 2004, 2005, people were routinely using this kind of sequencing apparatus, and I was also using my first sequence, this kind of machine. And then this kind of picture, and like that, and it was kind of work.
Yeah, online participants, please switch off your mic. Online participants are requested, please.
So it was taking almost a day to read the exact sequence. So this you read the sequence, and the sequence was transformed in this kind of architecture. From that, we will. So the sequencing machine or sequencing experiment contains these steps. You can make a photo, and I'm also going to give you a presentation. So we will see where one by one where you can have or you can encounter different problems. So let's see. You wanna make a photo? So here. Okay, so first let's see the basic principles of Sanger sequencing, and then I will run the video where some ideas will be clear. But as video is fast, I will try to make drawing over here, and then you will understand the video much better. Okay, so Sanger sequencing can be done by two ways. You can sequence PCR product, okay? So somebody asked me how big could be the PCR product. So 1000 maximum is the range. So if you have 1000 base pair, you should be able to sequence the full length. Okay? Or you can sequence the DNA as well, the whole DNA. But I recommend to go for PCR product. You can also sequence a plasmid. The plasmid DNA also you can sequence. So the input could be PCR product, input could be whole DNA, and input could be plasmid. But 99.9% PCR product. Okay, I'm going to draw the PCR. Okay, PCR cycle, that is denaturation, annual, and extension. Okay, so what you have, can you please tell me, apart from H2O, you have buffer, then okay, polymerase, GNTP, primer, and another primer. Okay, so this is a classical approach where one by one this will be denatured, so there will be two strands. The primer, forward primer will sit here, reverse primer will sit here. Then DNTP will come into picture. It will intermingle somewhere here, but with the help of polymerase, it will start to fill in and it will extend. Okay. This is the classical approach of PCR, right? So each and every component is used up in the classical PCR. Now we don't want a full length product, but we want randomly cut fragments. Okay, or we should be able to prepare or produce random length fragments, and that random length fragments could be done by adding ddNTP along with DNTP. Okay, ddNTP means a terminator. So ddNTP, I can write here. If the DNTP looks like this, okay, the ddNTP looks like this. Okay, so the chemical structure you can see. I don't want to spend time in that. So ddNTP is if it is T, normal T. After that, the polymerase can put here G, C, T, A, and so on. But when there is DD and TP, that means you have D and stop, D and stop, D and stop, D and stop. So mix of D and TP you add along with all components. Okay. So this is first change in classical PCR. You have the PCR, but you add DD and TP. Stop means there is no filling of next nucleotide. There is a stoppage. So what will happen? Your primer will sit, and then randomly, if there is G here, then DDNTP, DDNTP, if it sits, this fragment will fall apart. In second round, if there will be DDNTP sits here, this fragment will fall apart. Okay. If in the second round, if DDNTP will sit here and that is C, this fragment will fall apart. So there is a creation or generation of different fragments based on where your DDNTP will sit. Okay. Did I clear it very well or no? You are still confused. Okay, you will see the video as it okay. So thanks to combination of DNTP and DDNTP, there is extension and stoppage, extension and stoppage, extension and stoppage. And this reaction runs for 50 times. So trillions of the fragments, different fragments are generated. And DDNTP contains a fluorophore. Not only is degenerated, but it contains a fluorophore. Okay, different fluorophores. So there are 4 fluorophores. C, a T, usually we have red, C usually have black, G usually have, colors are different here, green and so on. Okay, so there are 4 fluorophores. So when there is a stoppage, the fragment always have some fluorophore. When there is stoppage, because of the DDNTP, there is always fluorophore attached. So you have millions and millions of the fragments with fluorophore at the end. Okay, and this reaction always and always run only in one direction, never in 2 directions. So you have PCR product, you have to purify this PCR product, and you have to send this to the company, right? Your PCR product. What company does is this reaction. So this reaction is also PCR reaction. Main difference is there is only single stranded PCR. You don't have there this kind. Okay, what they do is they take your PCR, they put your only forward or reverse primer, only forward or reverse primer. So what will happen is that with this sense fragment, you will have different antisense fragments of different length, like that. Okay, and these fragments you are going to analyze. This is the basic principle of Sanger chemistry. Okay, now ask me any question if you have.
One more component it will require is DNA. We need to add in that.
Yeah, DNA. What PCR product you send? They add PCR product here. Multiple fragments, because that fragments you are going to read, and based on that reading you have the ATCG sequence. You cannot read in one row. You cannot read in one row. Don't compare with NGS. NGS, for example, yesterday what we saw, the DNA is going through the nanopore protein, and it is reading the whole. In this chemistry, the DNA as such is not readable. DNA has to pass through the capillary or through the gel, and based on the length of the DNA, it will be separated, and then separation will be read. So you will see the video now, okay, and then we will come back to the questions.
First took place, but a sequencing reaction will take place. A mixture of ingredients added. These include free DNA bases A, C, G, T, DNA polymerase enzyme, and DNA primers. Modified DNA bases labeled with colored fluorescent tags are also added. These are called terminator bases.
This is the PCR product.
It is heated to -6 degrees Celsius. This separates it to two 6-strand. Temperature then lowered to 50 degrees.
Oh.
These include free DNA bases C, G, T, DNA enzyme, DNA primers. Modified DNA bases labeled with colored fluorescent tags are also added. These are called terminator bases.
Just a minute, we'll stop.
Mike,
Yeah, I turn it off. No, this mic is on. There are several mics on. You have to turn off this mic as. random process. Yeah. So there is no chance. There is no chance of missing any. Because the random process is so. So the random process is so great in this case that you don't miss any nucleotide actually. It may happen that you will miss. So do you remember my first lecture where we had runs and repeats? You remember? Okay. So runs and repeats means when you have AAAT, AAAT, AAAT. So it happens with AAAT only. So when there is AAA 10 or 13 time, then the polymerase will slip. It will not just add adenine again, but some adenines are not included. So in sequencing, those adenines you will not see. So only in this AATT, you will have some missing of some nucleotides, but rest is fine. All other. Uh, when you have non-specific, okay, so we are going to see the troubleshooting of that all. So principally, you should not send to company a sample with non-specific bands. You should send to company only specific. Sanger sequence is not able to sequence 2 or 3 samples at a time or 2 or 3 DNA at a time. Only one DNA species at a time. Okay, so that is wrong to send non-specific. Yeah.
That company said that when you are sending the sample, so which primer you have used, you should include that. So whether we can do that primer with other primer. Uh,
It depends on what you need
okay.
to sequence. For example, if you want to see only the genospecies, okay, so any of the primer is okay, or the forward or reverse primer is okay. But if you want to confirm the sequence and you want to put into the gene bank the whole length of the gene, in that case you have to have sequence from the forward as well as reverse, and then you have to combine forward and reverse into one sequence. I If you want, actually I'm going to show you tomorrow this one. Okay, so how to make the consensus of 2 different sequences and how to cut the bad part, how to edit the good part, and how to make one consistent sequence from both sequences. So tomorrow you are going to see. So if you just want to see what is that, any forward or reverse primer is okay. If you want to have confirmed sequence of any gene or any PCR product, you have to sequence from both, by both. Uh, I prefer reverse primer. If I want to see what is that, I prefer reverse primer. Um, more than 60-70% I get good results with reverse primer than forward. I don't know why, but for me reverse is lucky. Yes.
That there are different primers they are used for one species. So whether there is some unique location that should be sequenced, or when we do sequencing, so that some unique locations should be sequenced, or that anything is criteria is there for sequencing. Uh, Suppose some variant, any variant which is more pathogenic, virulence factor. Whether to target particular applicon or not. Yeah, that's like.
I understand you want to go check each part, want to check particular part with the
Yeah, yes.
that you have already included.
No, but sometimes there are different primers uh, for one organism only. Yeah, yeah.
Multiplex. Like you
Or,
want to know, like multiplex PCR you want to know, or different
no,
genes for one
like
pathogen?
some there are strains which are some pathogenic. So in that which like some there are again genes are there which are
Yeah. Suppose for listeriosis, there are 5 genes are there. Yes? Right? So we have to, yeah. in example.
Like
That example you can give from list
yeah,
hygiene are there.
yes, yes, that's what. त्याला काही क्रायटेरिया आहे का माझा तो हा ते तर करणार पण त्याच्यासाठी मग कोणता काही आधीच आपण सिलेक्ट करते वेळेस कोणता पार्ट ओके
If you can have some example.
Yeah, yeah, sir. You want to ask if you can give some examples.
Okay, so I think what I understood is if Listeria has two or three different genes with variations. Okay, so in any bacteria, there are housekeeping genes or the conserve gene and variable gene, or any organism, I could say. So whatever are the necessary for the normal function are the conservative. Okay, that you don't have to dig into that. For example, 16S RNA. Okay, 16S rRNA is highly conserved across the genera, and you can genotype based on 16S. You can sequence 16S, and you will end up with Listeria. Okay, so and now you want to go ahead, either Listeria monocytogenes or Listeria X, Listeria Y. So you have to go for the variable genes, and that variable genes are, for example, listeriocin or other surface protein coding genes. Okay, so OMPH, for example, porine is a conservative because porine is necessary for the function, although that is membrane. Then company will send you any results, and that could be bad. Okay, so do you remember from my first presentation the dimer, the self dimer or cross dimer or heterodimer? Yes. So this is a typical picture of dimer. Where are those dimer? Can anybody tell me? Here. Yeah, so these are the typical dimers, either self or heterodimers. So some people are lazy; they don't want to go for gel electrophoresis. Okay, and what they say, send to the company direct PCR product. So never send your PCR product without purification. So first rule is always purify your PCR product from the gel. Okay. So those who have not sent yet and want to send, you have to have agar gel electrophoresis, and after that agar gel electrophoresis, you have to send the sample. Uh, the wells comes with different sizes. They can come with 10 well, 20 well, 15 well comb. Okay, use the biggest well possible and put all 50 milliliter 50 microliter of the sample in one well and make the electrophoresis. So the concentration will be better. Now this is another typical example. Where is the dimer here? Can you predict where could be dimer here? So unfortunately this is a dimer. Unfortunately the bigger is a dimer and upper is a PCR product. Okay, so it may happen that your dimer is bigger than your PCR product. Bigger means brighter than your PCR product. So dimer is never below your PCR product. Dimer is never below your PCR product. So don't cut this whatever you see, but you have to cut this small. Okay.
That faint banding concentration of DNA is low, right? Also.
No, that we will cover in the next slide. The concentration of DNA. But this bright, yeah, bright indicates that your primers are making not self dimer, but like this dimer. So why like this? Not self dimer. If there is a self dimer or hairpin, what will happen? It will not grow at all. The concentration will not increase. But if forward and reverse will attach like this, the polymerase will work and it will fill. So you will have many copies of this, and that's why it's getting brighter and brighter and brighter. So difference between this, for example, and this dimer is this is not, for example, this is not a this is a heterodimer and this is a self dimer. Sorry, this is a heterodimer and this is self dimer. Okay. Okay, so this is a typical feature of less resolution of the gel, and your dimer will look like it has some 300 or 400 base pairs. Okay, so less resolution means, see, this is 500 base pair, 1000 base pair, and 5000 base pair. Okay, because of the ladder, I know what is what, and the dimer looks like 400 base pair. Yeah, this is 500 base pair and it looks like 400 base pair, but unfortunately this is dimer, and dimer comes up just because there is no resolution. Okay, the separation of the 500 base pair and down, what is wrong? So there is no good separation between the 500 and 100 base pair. So you think that there is some product you will cut and send and no result. Maybe chances. Maybe working. Connection is wrong. Okay, let's go ahead. Okay, now this is answer to your question, Vilas. This is the answer. So this is typical dimers, PCR product, genomic DNA. Okay, you have less genomic DNA and some middle concentration of genomic DNA. So as you see here, you put a lot of genomic DNA and still it remains here. So this indicates that you put a lot of genomic DNA, and that genomic DNA may inhibit your PCR reaction. Okay, so there is always a range of input, how much DNA you should add. You should not add really a lot of DNA. This is clear example of a lot of DNA. How much? Well, DNA you should not in nanograms always. So we add usually 1 picogram to 1 nanogram. 1 picogram to 1 nanogram. Okay, but we use high fidelity and very high efficient polymerases. You can go up to 10 to 50 nanograms, but never add 100 nanograms. Okay, now answer to your question, I guess. What if there are non-specific product? So whether company will be able to sequence non-specific product, and company will fail to produce any sequence out of that. So this is typical examples of non-specific. For example, this. Okay. For example, this, they are very close. The non-specific is very close. So how do you usually send sample for sequencing? Have you sent sample for sequencing? Have you cut from the gel? No? And that we are going to see why it is not there. Okay, but I recommend you all never send just PCR product. First, you will lose the money because company is going to charge you, and company will not tell you which product they sequence. Okay, a PCR product, but do you see in the gel or no? Yes, then why you don't cut and send it? It's very simple, because you can send this kind of PCR product, right? And company will ask which band I will cut, either this or this. Whether they ask you, never. In this case, you have this band, this band, this band. Which band should I cut? They will ask. They never ask you, right? So what they do, they just pick the brightest one, sequence, and send you the result. But that result might be not your result. That result might be a non-specific result, because you are going to add whole genome in the PCR reaction, and whatever is going to amplify, and whatever is this and this, and you may get these results as well, and you may not get these results. Okay, so send always your sample, cut yourself by yourself, and then send it. Never send. And cutting takes 1 minute, purification takes 10 minutes. That's it. Okay, and how to improve the purification is very easy. It's not that difficult at all. And the columns which you use, actually we produce our own columns. I call my diploma students and they prepare their own columns, and they prepare columns for whole year. So we prepare our own column. It is not expensive at all, and it is not difficult at all. Okay, if you have used column, you just recycle them and use it. Okay, so it is very easy to purify. If you don't have columns, you can put ethanol and send it. Okay, so ethanol precipitation is also possible.
Yeah, sir, just one question I want to know, because in first picture,
Yeah.
the sample number, well number, because first is the ladder and then fifth one. Fifth number.
Yes.
Because there are three bands are there. So in that top one is the, I think that is the required one, means specific one. And yeah, and that one is the non-specific one.
Yes.
And below is the dimer.
Timer.
Yeah. So we need to cut that specific one,
Yes.
and that we have to set. Yeah.
Yes.
Because
Yes.
always for comparison we require the labeling, means ladder,
Yes, yes, yes.
specific bands. Yeah.
Yes. So unfortunately, company will not do it for you, because company never knows what product you want. Okay, so you have to cut. Okay, and this is a typical unfortunate result. For example, if you compare this result, at least Vilas was able to say that this could be a specific. Now tell me which is specific here. It is not possible to say. Yes, but in this case, for example, your predicted product size is 1500, and this is 1400 something. So you never know which is your. Yeah. So in this case, I do not recommend to send PCR product. Always cut the band. And to your question, Rashmi, low concentration of PCR product, what should I do? So there are two options. First, make the purification better, or increase the concentration of PCR. Okay, that means you can do 3 or 4 times PCR. Make put together and purify. Okay. Yes. How to fix? What does it mean? Fix? Yeah. How to fix? So which is the better one? So you start in the middle somewhere. So 100 picogram, for example, you can. How to make? Dilute it. You, you, you have to quantify the DNA by spectrophotometer, nanodrop or whatever spectrophotometer, and then you have to dilute it. If the concentration is, for example, 100, you dilute it 1000 times, and it will become 10 pico or something. Primer dimer with what? DNA. Mere.
I think question is also in that, answer is there.
Yeah, actually, smear DNA or smear whatever you get there are again 2 or 3 causes. First one, your DNA is degraded by your hands. So, for example, main thing is rice. If you eat rice and dal with, of course, with the hand, and rice has a lot of these DNases by default. And then with the same hands, you start manipulating your DNA, and then it's done. So never manipulate the DNA with bare hands, but always hand gloves. Uh, and there are several other plastic wares that has DNases. So you need to use DNase free plasticware always. Second thing, and the smear. Smear could happen also because your buffer system is not good in the electrophoresis. Yeah. So PCR never makes the smear. Okay. PCR is either 0 or yes. So PCR is never something. No. PCR technique is yes or no technique. It will never give you smear like thing. Smear is either in the DNA or in your gel, and that is a buffer system. And buffer, if it is old, agarose, if it is old, you will have smear. Or if you run the uh, electrophoresis very fast, you can have smear. So there are several other, but PCR never makes the smear. I mean PCR reaction. Okay, so this is another example of low concentration of PCR product. So can you see here is quite low concentration, and what I recommend in this case is to make multiple PCR reactions. There is another very amazing trick. Okay, sorry. Uh, Several people, they may go for a second round of PCR. Yeah. So what you do is you purify this or no? You put the PCR product. Okay. So I don't recommend that, because what will happen is, for example, in this case, you have PCRs and you have several bands over here. Okay. And if you take the
अभी क्या है सेम जो है ना इनका बेच के साथ है राजू
Hello,
सेम कंडीशन
online participants.
अभी क्या इनका
Please,
बेच बहुत
please
बहुत
mute
बहुत
your mic. Online participants, please.
So I don't recommend to use PCR to PCR or amplification of your PCR product, because a lot of garbage is there. Garbage in terms of non-specific products. So this is typical example. You have several, and all these several non-specific bands are going to amplify in the second. Okay. So the best way is take 200 microliter tip. Okay, 200 microliter tip, cut it a bit. Yeah, cut, and then you have a 10 microliter long, or whatever you have needle, for example. Okay, and the idea now is you see the gel. Okay, put the gel on the glass or wherever on the transluminator, and you are seeing that band, and punch the tip into that band. Okay, punch the tip into that band, and exact band. So I will punch here, not here. I want to amplify this, so I will punch here, and then I will release whatever material is came into the master mix. So don't worry about the agarose, don't worry about anything, and you will have amazing result of a big prominent band, amplified of this band, not this, not this. Somebody is doing that? Look, yeah. Can you bring the 200 microliter tip? Yeah. आणि कात्री पण आण, निडेल आण. Yes. Yes. Of course. to take that gel out, because with the pipette you are not able to take out that gel. So gel will just entangle in that tip. Okay, and you have to release the gel. So you just put tip, yes, gel, and release that gel into the master mix. Sorry, tip and needle combination. Yeah. How to use it? कळलं? No. So how to increase the concentration of PCR product, a specific PCR product? Because if you do a lot of PCR cycles, again the results will be like this. Okay, non-specific results. You do reamplification, the result will be the same. So the results are not going. Wow. Take this back. So never use this with the filter. You can use without filter. So it depends how big you need. Okay. So for example, like this, and make the needle blunt. So actually I cut the needle as well. Okay, so now the needle is blunt. Okay, needle is blunt, and what you have to do, you have to use the long needle, not small like this. And what you have to do is you have to just put, yeah. So the idea is, if this is gel on transluminator, and if this is a band, yellow band, I will punch it, right? Do you remember Aucherloni gel diffusion test? Puncher, yeah, it's the same. Yeah, the same. So I will punch it on that band. I will take it. Well, I I will punch it first. Okay. So gel is here now, and then I have a master mix tube, and I will release this gel, whatever came into that master mix, and I will run the PCR. No interference of all agarose. Okay, and then if somebody will try, then send me the picture of your result. Okay, and you will never be sad with this. Okay, now which gel system and which dye system you use? Cyber green or ethylene bromide? Really? All ethylene bromide and UV? Crazy. Yeah, because actually now ethylene bromide is banned for the DNA purpose. We cannot use ethylene bromide. So I think you are using the hand gloves and everything that is extremely carcinogenic, and you should not use ethylene bromide. Okay? So you should use cyber gold. It's extremely cheap, cyber gold. So you can buy one 10,000x cyber gold from, it was in Vitrogen, 10,000x cyber gold. And I guess that uh, you are staining your gel after the electrophoresis, right? While preparing the gel. Okay. So what I'm using, or we have invented way back, is put the dye in the sample, not in the gel. Yes, in 6 select. And then we developed the machine with lead strips from the market. So when lead started to come into market, I purchased blue lead for 10 rupee or 10 euro. And then with the different dyes, I was trying which dye is good with the lead. And I found that cyber gold is the best with blue lead strip. दुकान पे रहती है ना lead strip. So I use lead strip with cyber gold. So I will tell you advantages and disadvantages of that later. Is extremely important where you are losing your DNA. So this is a typical picture of the good PCR product, which has some smear, but that smear you have to remove from by cutting your PCR product from the gel. Okay. So let's go to gel purification. So whatever we saw is different problems in the PCR, either non-specific, low PCR, dimers, how that could affect our gel concentration or PCR product, and how to re-amplify our PCR product. Okay. So if you have any questions, then I will take later. I will go to gel purification. This is quite lengthy. I will check if it is really lengthy, then I will cut this video short. Actually, this is question. As I'm going to send you, I hope that you have made column-based purifications. Are you aware with that? So okay, so the first step of the gel extraction is you cut the gel by scalpel. We use normally surgery scalpels for that. But main thing is that you have to burn the scalpel before cutting. So what you have to do is you have to put into the ethanol and burn it. So all the DNAs and everything will go away. No cross contamination. And this is the cutting of the gel, and you cut the gel band and put into the normal tube, and you weigh it. So how many grams you have? Usually we have 0.1 gram or 100 milligrams. We have a gel band. Do not make bigger gel bands because that will affect your concentration. Okay, so the gel should be around 100 milligrams, 0.1 gram. Never go beyond that. Okay, here you see the 400 milligrams total weight, but what I try is I try to remove all the parts which are not fluorescent. Okay, so and then I cut it short and make only that dense part, and that is giving me very good result. Okay, so okay, stop here. So this solution is QG buffer. Okay, so the QG buffer, or different companies they can say QG buffer or binding buffer or solubilization buffer. There are different names. But the first buffer you are going to add in the gel contains chiotropic salts. Okay, so guanidine thiocyanide. Okay, as this is toxic, you have to be very careful by handling. It is thiocyanide, and that will disintegrate your agarose. Okay, the main thing is that addition of guanidine thiocyanide should not be less. You can add more, you should not add less. For example, if you have 100 milligram, never add only 100 milligram, always minimum 2 times more. Okay, otherwise your gel will not be solubilized, and everything will be lost in unsolubilized gel. Okay, so this is very important step. pH, I don't think you will have problem with them. If you make normal buffer and gel, you will never have problem with the pH. I will skip that. Can you stop? So the binding buffer they have added 2 times. First to solubilize gel, and here what you saw is they added once more. So addition of once more binding buffer is to just purify your DNA. Water, gel or agarose is remaining because of the second round of purification. If you addition, your gel, whatever remaining on the filter will go off. Okay. This is washed. Okay, please do. So in this, in washing step, there are two things. First one is concentration of ethanol. You have to have ethanol in your washing, because do you know what membrane is this? What kind of membrane is this? Hmm? Okay, so this is silica type B. Okay, this is silica, but this is type B. There are around 20 different silica, different types, but only B type will bind DNA. Okay, not all. You can purchase B, that is really cheap. I recommend you to purchase from Merck. It is previously PAL Corporation. Now it is Merck. So silica type B you can purchase and you can make your own columns. So this filter is nothing but very small piece of silica type B filter paper. Okay, and that filter paper has inherent capacity or ability to bind the DNA in presence of salt or ethanol. Okay, I repeat, in presence of salt or ethanol. So in the first case, in the binding buffer, we have high concentration of chiotropic salt, that is guanidine thiocyanide, right? So in presence of salt, DNA bound to the filter. Now we want to take out the salt because we want DNA very pure. So the best way is to remove the salt is to put the ethanol. So in the washing buffer, you have to add the ethanol at least 70%. Okay, so you can make your own washing buffer, either TE buffer with 70% ethanol, or you can use 70% ethanol, no problem, if you don't have wash buffer. Okay, so in presence of salt and ethanol, DNA is going to bind silica B, and if you remove that ethanol, in water is going to release. So it's very simple principle. Okay. Okay, so this is here. You have pure DNA. I will stop here. Thank you. And one last recommendation in purification step is, first is binding, second is washing, and then is elution. So after washing, you have to spin your column at least 2 times dry. That means you have to remove all the stresses of ethanol. Otherwise, you will not have good elution. Okay, so just spin it twice. Twice means at 1000 RPM. Just press button and then column will spin for 1 minute, and that will remove all your ethanol traces. Okay, so the purification step looks very easy. It is easy, but there are several problems. The kit comes from Quagen or other companies, but ethanol unfortunately comes from you. Okay, you have to add ethanol in the washing buffer, and that's the main problem. These are the steps: binding, wash, elution, and in the washing buffer you have to add ethanol. Do not add ethanol from any company with low quality. Always molecular grade, highly pure ethanol. Invest some money in that, but result will be good. Okay, then this video here tells you how to cut the band, but it is a bit long, so I will just keep, and as I'm going to give you this, you can see at home. Okay, now where you are losing your DNA, Rashmi, the main problem. How many seconds you are cutting your band? Yeah, that's a lot. Okay, so do you know why more are the seconds you are losing your DNA? No. Okay, So all those who are using the UV as transluminator, you are burning your DNA. Okay. Now, just when you will be back, do one thing. You put the gel, turn on the UV, and keep for 1 minute and see if your band is there or no. Okay. So your band will vanish, or the concentration or the intensity is going down. Now you have, for example, 1, 2, 3, 4, 5, 6, 7, 8 bands, okay, on a gel. You are putting on the transluminator, you are cutting here, this band is exposed, this band is exposed, all bands are exposed. So for example, this band you cut in 10-15 seconds, but unfortunately this will be exposed, and so on. So by the time you come over here, it is gone. Okay. So I recommend you do not use UV. Change your transluminators. Okay, and it's very easy. I will show you our own transluminator.
इसमें आज ऐड नहीं हुए क्या? इसमें ज्वाइन कर रखा है नहीं? क्या है ये जो?
Can I? Okay, so this is our own transluminator, homemade. What you see here is just a plastic cover. We found some very transparent plastic cover, and under the plastic, what I have put is stuck. Let's wait. Regular. Okay, whatever. So this is just plastic box, and under that plastic box inside I have put the blue LED lights, strip of blue LED light. Blue nanometer is 488 nanometer. You can see the back side of the strip, the nanometer. So as the LED light has a specific wavelength, LED light, each color has its own specific wavelength. So if we have normal light, it has mix of all wavelength, and we need a cube to separate those wavelengths or those light. But if you purchase the LED light, LED, we say LED is LED. So LED light strips. So each color has its own wavelength. Okay. If you purchase the blue, it is 488 or 480, and that is not harmful for DNA at all. Okay, I will. Yes, it's charging. Okay, I will check if we will see later. And this is a way how we cut. We put on the lid, and we put in this side, we put some paper and we cut one by one. Although we are using blue light, we protect our DNA from the light, because all the biological molecules or all molecules are sensitive to color. Some are light, so some are faster, some are slower. For example, UV, it will degrade the faster. Blue light will degrade slower, but still it will degrade. So we try to avoid that degradation, and we keep, if there are 1, 2, 3, 4, 5, 6, 7 different bands, so we
Online participants, please mute your mic. Mr. Dr. Said.
Okay, so do you understand how to make that blue strip? If no, then I'm going to send you this, so you can see the video. Okay, let's see how to increase now concentration. The main thing is that you have to remove the excess gel. Okay, so this is your gel band. Okay, what you see, and in that gel band, what you see is this is your DNA, and this portion is only gel, and you have to remove that by just making a surgery a bit. So you have to remove that gel and keep only this and purify only this, and then you will have greater chances of having good concentration of your PCR product purified. Okay, I will skip this. Okay, now let's go to the buffer system. How buffer system will affect your concentration? There are two buffer systems, TBE and TAE, right? This borate is common what you use, and TAE buffer, TIE, we say TIE buffer system, and we don't use any of those. They are very expensive and very difficult to produce or prepare. The TAE is one of the quite difficult to produce, and they are very slow. They are very slow. I have only 10 minutes. Okay, so how many minutes you run? 1 hour? Half an hour? I want 10 minutes, for example. Okay, and I would like to see how it is separating real time. I don't want to wait and say, okay, and then I will put. So what I do is on that transilluminator, which is blue, I put my tank on that, and I use the borax buffer. So this is sodium borate. This is boric acid. It's extremely cheap. Boric acid you can purchase anywhere. Okay, so use, but don't purchase from the from the market. So this is borax. 20 gram of borax in 1 liter is really cheap. Only thing you have to is not tris borate. Okay, this is boric acid. No, not this. No, this is different. This is borax. This is borate. This is borax. Now you will see the parameters over here. 200 volts minimum. You can go to 300 volts. Okay, normal volts are 180 or 120 is normal. You run 180 volts. We run minimum of 200 volts. And when we compare the recovery, it is more than TB and TIE, because it directly influence on your gel bind or DNA binding to the membrane. Okay. Okay, so we saw the buffer system, quantification and purity check. I hope that you use the nano drop or any spectrophotometer for purification. And what contaminations you can have EDTA. If you use TAE or TBE, there you have EDTA. If you use borax, you don't have EDTA. Okay, so this contamination is off. Guanidine salt. So guanidine comes from guanidine thiocyanide. Okay, so you have to wash your column vigorously or 2 or 3 times. You have contamination of oligosaccharides. It comes from the gel itself. You have proteins. Proteins come from DNA polymerase. It's protein. Okay, Taq polymerase. Silica beads. Silica beads come from the membrane itself, and so on. So all these contaminants, they will come during your purification. This is a typical picture of a very good DNA with good concentration. You have to see this value, this value, and this value, and this value. And based on these 3 values, you have 1.8 and 1.59. So purity of your DNA should be 1.8 minimum. 1.8. It should be purity. If it is 1.5, don't send sample for sequencing. So for DNA, what you are going to send for sequencing, the purity should be 1.8, and the concentration should be at least 5 nanogram per microliter. 5 nanogram. So Rashmi, when you are saying that the concentration is not good, good means how much, right? So good means if you have 5 nanogram per microliter, it's okay. $600. Why? Huh? You are saying for DNA, not PCR product.
No, sir, for DNA.
DNA 600 is okay.
We used to get that way.
No, here we are covering the PCR product.
The PCR, sir, we always got around 100. That was our
It's
number.
too much.
We used to get 100.
It's good. Oh, we never get 100.
We got because we had to write that in our thesis, so I clicked all the pictures.
Okay, so if you have 5 nanograms, is very good. Okay, so then there are different issues over here. See, this value is 1.9. Okay, you will see, oh, my DNA is very good because it is above 1.8, but this value is 0.08 only. So if you compare this value, you have 1.8 versus 1.5. This value is quite different. Okay, 0.08 only, and that gives me some hint about there is some contamination, and that contamination is because of guanidine contamination or no DNA at all. Okay, so mainly there is guanidine contamination. Okay, so then there is another, you can see here 1.5 only. So you can compare this graph versus this graph. It's completely different. 1.5, although it is telling me that 32, but I should not take into consideration 32 because it is not. The concentration is purity is 1.5 only. This purity sucks, so I should not send sample for the sequencing. Okay, this is typical picture of protein contamination. That means the Taq polymerase is there still and you have not purified, and that Taq polymerase will kill the next Sanger sequencing. So this is typical picture. What you saw first is like this, yeah, but this curve is like this. So this is typical of protein contamination. And this picture is again, now you can compare this. Okay, for example, what I said, you have contamination over here, the concentration is 32, but see the graph, it is still okay. This graph is not dented. Here you see the graph is dented. It's like that. That means silica has come from the filter. So your filters are old. Okay, silica is old. You can avoid this contamination because silica is it has weight. Okay, it is not that light. It's denser. So you just centrifuge at 10,000 rpm. Silica will sit down. You take upper part, and your sample is free of contamination. Okay. So this is more or less in your lab. I mean, when you send sample to us or to a company, what things you should consider. And then next part is what can go wrong in our lab, I mean, or a company. So there are also several things. We can take a break over here and continue that part later. Any questions still on? So far, no questions. Good. Sorry?
Sir, may I ask you one question?
Yes, yes.
Thank you, sir. Thank you very much. Sir, you said you can reuse the column
Yes.
for gel purification. How can we use that column? Is there any method specific for that?
Yeah, there is a treatment, but unfortunately, I can send you all the protocol how to reuse the column. There is treatment. You can use HCl or NaOH to kill the DNA. So strong acid. The problem in that is your silica is going to get fragile. Do you have used columns, Rashmi? You have used columns? Sorry?
Yes, sir. We have used the column.
But do you have column here? Yeah, can you bring column? Okay, so when we were trying to recycle the column, and we found that it is not good idea for after 2 or 3 cleaning is not good idea. So what we found is the best way is you purchase the filter, that is silica B, and it is, I think, 20 euros. With how much it will? 2000 rupee only, and it will last lifelong. It is lot of filters you will, huge quantity of filters you will get. And then what you need to do is you need to have just some punching device, just punch it and put that in the column. So I will show you how to disintegrate or dismantle that column and how to put that, and yeah, and it is new again. Okay, so I better recommend you to recycle in that way, that you change the filter, and filter can be purchased in 2000, and you cannot purchase whole what I can say, whole kit in 2000 rupees. Nope. Yeah, it's quite expensive. Yes. Okay.
Thank you very much, sir.
Yeah, welcome. You put, in case of multiplex PCR, first you put on the gel and then cut from the gel, separate on the gel.
For the sequencing, do we need to pull that code and
From the multiplex? No, never pull, because each product is different. So you have to cut the band and treat as a one sample, another band, another sample. Yes. Otherwise, there is no sequencing. Okay. For synthetic, okay, let's have a tea and. Okay, I don't teach this actually, but that is my hobby. And as we do a lot of production and like self-sustainability or something, although we have a lot of money, I always teach my students, make yourself all this. So you will understand, and then when you will go back, you will open your own company. So that's why we try all this.
What about that? What about that chemicals and buffers?
We have a.
We can make it.
Yeah, you can prepare your own thing.
Whatever that is there in.
Look, you need for purification. You need agarose. Yeah, agarose is cheaper from ABT company, which is Spanish company, but ABT is the most cheapest agarose and very good. You don't purchase from Coagen or Biorat. Very difficult. Yeah. Yeah, and we recycle the agarose. We recycle the agarose. That means. Okay. Look, this is your agarose. Right? Yes. Okay, first I will tell how to recycle. I think you will open your own laboratory now. Okay, agarose is one of the expensive part in the electrophoresis and everything. That's why several people they say I don't want to make this. So the trick is that you cut this, and you cut this, you cut this. Okay, and if you don't have this kind of, you just take this part and dampen, for example, big box. Okay, and put it in freeze for lifelong maybe. And for example, you want to recycle now the agarose. Okay, so you take that, you boil it. Okay, it will get melted soon, and then you just pour that into slabs. Okay, in one plate, and you cut the slab. Now, for example, you cut the slab, and you know that this slab corresponds to 200 milliliter. Okay, and stack the slabs. Okay, and that slabs can remain in the freeze several years, but minus 20. Okay, minus 20 not. Okay, and then what I am going to do, I want 200 milliliter of the agarose. I will take that slab, one slab, I will cut, I will put into the micro oven, and gel is there. Yeah, but you can have this contamination. So I just cut this band and throw small piece. I just cut here, throw away. And you may have sometime this one. So I also tried to cut out genomic DNA and the ladder. So I take the only, but still it is a lot of recycling. Okay. Yeah, also. So you also recycle. Okay. Yeah, resolution affects. Okay, let's have tea and yes. 10 minutes. I can send it. I can answer later.
There is an instruction for online participants. Now there will be a 10 minutes break and the remaining session will resume afterwards. Thank you.
ड्यूटी जिसमें दिया क्या है ना उसकी जमी और गई उसकी
बुखार का थोड़ा सीजन चल रहा है वो छोटे वाले बेटे का 1 महीने पहले तो होया था। ई पेपर देगा है स्कूल में ही फोन आया
All the online participants are requested to join the session.
Down here, that's plastic filter, plastic. Some companies Biden more filters you have more, but believe me, one filter is don't put three eyes. What will happen is stop. And once you cut this, what you have to do? Punch, sharp punch that filter paper. Why? Just insert here that filter paper. Example, that O-ring goes back. That's it. Filter is ready. And this filter you just wash 4 times with water. This
Plain water.
Huh?
Plain water.
Normal liquid water. And this tube also you can recycle. So at the end you don't need to buy any plastic at all. Buy only filter and recycle. You don't have to sterilize. Why you need to autoclave? No. You don't autoclave at all. You don't need to autoclave because no autoclave. If you want to clean it, don't autoclave, just put in NaOH. Because the plastic filter which is here, the white plastic filter is here, is the same plastic filter what we use for chromatography, the column chromatography. And we use NaOH 0.5 molar of the NaOH. So we use 0.5 molar, okay, not millimole, mole. 0.5 mole of NaOH we use in the HPLC or FPLC to clean the column. And once you put NaOH, everything is clean. Yeah? And the settle time is around half an hour. So let NaOH sits half an hour. Everything is closed. Then you have to put it at least 1 or 2 days in ample of water, but normal water, tap water. Okay, then put it in the clean distilled water, then it's fine. But usually you don't need to do anything. You just clean this with some water, that's it. Okay. Wash it, yeah. Electricity आहे इथं. Yeah. Okay. Hello? Hello? Offline participants, do you hear me? Offline. Or online, yes. Actually, offline is not a word for us, is in presence or online. I'm get jumping. Let's see. Let's see if in 1 minute, if video is not lengthy, but let's see if you will have luck. Okay, so let's go. Okay, so till now we saw that what could be the problems in your laboratories, how to tackle with those problems, and now we can see company is not always right. Company can make some problems, and company can fail the Sanger sequencing. So if you follow all the instructions, and if you send your DNA pure, okay, 1.8, very good peak, you cut only single band, there is no primer dimers and so on. So 99.9% you're on right side. Okay, now if you don't get the sequencing, then the problem is at company side. For example, as we are doing sequencing commercially as well in our lab, we give service is free of charge service to our clients, that we also guide them how to make good sample. And if there is some problem in our side, I also personally I check in the evening or some whenever I have time, I check where could be problem, because that gives me feedback that if my machines are working very good or not. And it is also my hobby to maintain the machine. I have two technicians, both are girls, and they are really amazing to maintain the machine. But if something there is wrong in the capillary or something, then I accompany them to clean the machine and so on. So there could be a lot of problems in the machine or the Sanger chemistry as well. So this is what you get several times, that you don't have sequence and you have penta n or 5 time n. That means we say samples were wrong or sequencing didn't work. Now you see the difference between this and this is actually don't see by naked eye as you are not expert, but what I can say is this is very poor quality of DNA. So either DNA is mixed, there are different bands, okay, and we will see one by one, or wrong primer was sent. So you have to keep in mind that you have to send the same primers while you are sending the sample which you use for PCR. Never. And that is big problem when people do nested PCR. Okay, because you have outer primers, you have inner primers, and you don't know which primer I have to send. Always send inner primer, never outer primer. Okay, but mistake may happen that you send outer primer and the reaction fails. So poor quality of DNA, wrong primer, bad water for elution. Now for elution, what do you use? Elution buffer, elution from the column. What do you use? Simple water. You need to use simple water. Never use TE buffer or elution buffer which company gives you. Never use DNA, RNA free water from the company. Never use your own water, distilled water. No. Or this could happen because of your primers are not good. So if you use freeze thaw, freeze thaw, freeze thaw cycles of the primers, it may happen. And okay, so this is on your side, and this is a company side, because the sequencing machinery is called as 3.1 big die terminator kit. And if it is old, you may have this, or the capillary is blocked. Okay, so the capillary is blocked. How capillary could be blocked? So capillary actually has 50 centimeters. 50 centimeters. Have you seen capillary of sequencing? Really?
स्क्रीनिंग कमेटी का मीटिंग में परसों पहुंचूंगा.
Online participants, please mute your mic.
Okay, and what is capacity of that capillary? How many microliters of the gel? Because there should be some gel through which the DNA is passing. How many microliters could be in one capillary? Can you guess? Only 2 microliters of the gel in one capillary. Only 2 microliters. Okay. So this capillary is like hair, is hollow inside, and that inside stuff is filled with 2 microliters of, from this end to this end, only 2 microliters. So it's very easy to get clogged. Okay. So this capillary could be blocked. Another issue, you may get this kind of result. You may say, wow, this is really good result. I have very good sequence. I can see ATCG, but sequence are bad because you have several multiple bands. Okay. So for example, you see here you have 2, and everywhere there are 2 actually. And you will get this result when you do not purify through the gel. And if you have a multiple bands, I'm going to send you, okay, you have multiple band in PCR, okay, or even you have non-specific band, then you will get this result, mixed chromatogram. And as a no gel purification and direct PCR, if you send direct PCR product, this may happen. That means there are 2 or more type of PCR product in sequencing reaction. It may happen. It is not rare. It is quite frequent. Okay, it is quite frequent. If you give a PCR, so this problem starts from PCR. You purchase forward and reverse primer, right? And you give it to your student, and student make the master mix, right? And they are new, they are young, and they forget to change the tip always. What they can do is they can make 0.6 or 0 or 1 microliter of the forward primer with the same tip, 1 microliter of the reverse primer, and there is then it start to get mix of both primers. Now we saw the basic principle of sequencing is to sequence only by 1 primer, not 2 primers, only 1 primer. And now you are going to send that mix, unfortunately created mix, to the company for sequencing. And always you will have this kind of mix reaction. Now, the mix is not equimol, right? Because your student has taken forward first, then reverse first, and she is doing routinely this kind of mixing. So this mixing is not equimol. This is just contamination. So what will happen? You will have very good sequence, and intermittently you will have that mixture. Okay, so this is a primer contamination, forward and reverse, pipetted with the same tip. Aliquots. Aliquots. No. Aliquots. Make aliquots. or minus 20. Yeah, for long, but tens of 15 years it can survive in minus 20. Actually, primers comes in mole, right? So you can have 600 to 700 microliters of the stock concentration volume, and then you again working to 10. So at the end you end up with 6 milliliter. And if you make stock aliquots, it's fine. So working could remain 6 months in freeze refrigerator minus 4 degrees Celsius. Yes, working. Then discard. Yes, then discard. 6 months. 6 months. Yeah, but it's up to you if you want forward or reverse. They can ask, but they ask to make the money. They don't ask to help you. No, no, no. They ask for money only because look, one sample means forward sample, reverse sample. Okay, and the company have trick to make money that oh the sample has to sequence by both. It's not true. You actually you can sequence only by one strand if you want to confirm by both strand. But if you say oh I want only reverse, you can send only reverse. They need only one primer. Okay, so this is fortunately is not your issue or your problem, but is a side of company. So this is typical blob formation. So this may happen because when there is Sanger sequencing chemistry, whatever DDNTP and whatever you saw, so after that I need to purify the sample with ethanol purification or other techniques to be able to put into electrophoresis. So I need to take out all these polymerase and all other things because then it will stuck into the capillary. Okay, so I need to remove all. If I don't remove in a company, then you will have this kind of blocks. Okay, so when you will see this, that means company have made a mistake. Okay, so this is, for example, you have a high peak. First 2 or 3 nucleotides are very intense, and then you lose the signal. That means the company has put a lot of DNA or very less DNA in cycle sequencing. It is not your problem, it is company problem. Okay, so this is classical example of primer dimer. So when the primer tend to make a dimer, okay, so you will see here a primer dimer stuff. So that's why you have double peaks. But then when the primer dimer is finished, you have very nice peaks. So typical primer dimer sequencing. Okay, so this example is either you have used error-prone polymerase, not Taq polymerase, or that Taq polymerase is not good. Whatever you are using Taq polymerase is error-prone polymerase. Okay, so whenever you are purchasing Taq polymerase, you have to check what is the error capacity or error of making things of that polymerase. So this is could be one problem, and one can be indel. What is, you know what is indel? Insertion and deletion in the gene. So when there is insertion and deletion in the gene, you can have one sequence, same gene, but there is a frame shift. Okay. Now the frame shift, you can see one sequence is this one and another sequence is now mixed up with the sequence. So that could be because of the indel. Company. Yes. Yes. Uh, mm, yeah, they can recommend you the PFU, but PFU is extremely expensive. Yeah. And if you use very good Taq polymerase, that is sufficient actually. Nowadays, you have 10 raised to minus 7 or 10 raised to minus 8 error efficiency, or so 10 what is 10 raised to minus 8 error? That means every 10 every one error after million base pairs. And if you have 400 base pair, how much will be error? So there is no error actually when we are talking about 1000 base pair. Okay, so don't worry about Taq polymerases. They company can insist use PFU, but you don't need to spend money in that. Okay, again let's go back to the basic runs and repeats. Okay, so this is classical example of runs, not repeat, but runs. Repeat means AAAT, AAAT. So AAAT is repeating. This is run. G is run. So once you have in your gene or PCR sequence, you have this kind of sequence G, then polymerase is doesn't know what to do. It stops. Okay, so this happens in the cycle sequencing. Actually, I mean, you have PCR product very well, but cycle sequencing has its own PCR, right? And that cycle sequencing PCR, unfortunately, something weird is there, so it stops. So this is a run stoppage, and this is called as hard stop. So when you are designing your primer, try to avoid this kind of runs in your PCR product. And then this is example of repeats, or as well run, but what you have here a, and then again the problem is that this is called as AT run slippage. So for example, you have here a T and then A, so then this part is problematic. So there is no stoppage like GGG, what you see here, no hard stop, but then it is just slippage, and then there is mix of. So it is not your problem. You send your sample very good. Company did its best, but the problem is in the nature. And what you see here is a ski scope. That is once you zip your sequence, you see that sequence is going drastically down, and the quality of sequence is reduced. So don't store your PCR product for longer time, maximum half month, 15 days, or you have a lot of salt in your DNA or different contaminations. So this is typical picture of old PCR product, what you are going to send to company. Okay, so let's have some conclusion out of that. How to avoid the bad sequencing. First is download this presentation and read carefully, because I have tried my best to summarize all the possibilities, and you also go through the videos. Always use molecular grade chemicals, don't use any cheap one. A third one and very important point is that never use UV transilluminator. So when you will go back, try to find out. Now companies are selling blue light transluminator, blue light transluminator. Okay, so use that blue light because UV is going to burn your sample. And if you are going to use that for cloning, I think nobody is using cloning, but if in future you want to do that, so UV is mutagenic. Okay, ultraviolet is mutagenic and it will mutate your DNA. So in cloning, you will carry over your mutation. Okay. Always gel purify the PCR. Never send your sample directly PCR product. For me, blue light and borax buffer combination is the best. Use good quality agarose. So I recommend you to purchase ABT company. ABT agarose is company, and I have awesome 10-15 years I'm using. It's cheap, but it's good quality. Always use DNAs and RNAs free tubes and tips. Okay. Uh, if you can't buy, if you don't have that much money, the simple trick is make a freeze thaw of your tips. So put at -80. Okay, put at 50 degrees Celsius, make 2 or 3 times, and DNAs will be degraded because DNA is also enzyme or protein. It has to degrade by freeze thaw. You cannot use, unfortunately, DPEC water, the DEPC water, but freeze thaw could also help. And always send pure and good quality DNA to company. Yes, but unfortunately, filter get clogged in autoclaving. So freeze thaw is still usable. Okay, so I had here coffee break. So we have some questions over here. For example, is screen not available for presenter? Four degrees, okay. Refrigerator is okay. Where to download this presentation? Okay, so I'm going to give you Rajpal, you. So you can share, or Vaidya can share all of my presentations. You can share, or I can upload in my cloud, and from that cloud you can download. I can send you on the link.
Sir.
Okay. Yes.
Hello. Sir, you said that ECBR is a ban, and you suggested cyber gold for using it. So, what concentration should be used in the gel, or what is the method to use this hyper gold? Am I audible, sir?
Do you see the whiteboard now?
No, no, sir.
Wait. Okay, do you see the whiteboard now? He sees the, okay. So I will give you some more information of how to use the cyber gold, okay? And that is quite tricky. So cyber gold.
No, sir.
Okay. So cyber gold comes with 10,000x. Okay. So what you have to do is this 10,000x from in vitro gen, you have to put 0. Sorry, 0.2 microliters of cyber gold. 0.2, never add more. Okay, 0.2 microliter of cyber gold in 6x buffer die or die buffer.
4x.
No, 6x die buffer, loading buffer, loading die, not buffer, sorry, but is
Oh,
loading
okay,
die,
sir.
gel loading die, 6x gel loading die, gel die, and that should be 400 microliters of gel loading die. So add 0.2 microliters of 10,000 x cyber gold in 400 microliters of gel loading dye. Keep it at refrigerator for 4 to 5 months, up to 6 months, no problem. Wait. Okay, so you have this stock solution of the dye. 1 microliter of this into 5 microliter of sample, DNA sample or PCR product. So mix on the parafilm. I use parafilm for that. So on the parafilm, what I do, I mean my student, they first put the spot 1 or 2 microliter, or I can go 2 microliter and 10 microliter of sample, or 10 microliter plus 50 microliter of sample is the same, 1:5. Okay, so you mix well and load on the in the well. Now, when you are making gel, don't put anything. No dye should be put in the gel. Simple gel. So you prepare the gel without any dye. No need. No, you don't need ethylene bromide. So cyber gold is extremely safe dye. It is not carcinogenic, and you put only 0.2 microliter in 400 microliter, and you use only 1 microliter of that. So if your group will purchase 1 cyber gold for all of you, and it comes actually 500 microliter of cyber gold. So this comes 500 this comes 500 microliter of cyber gold. So hectoliters of dye you can prepare. So if one of you buy, then you just share with other, and is enough. Okay. So the big advantage of this system is safe. Okay, it works with UV, also it works with blue light, and blue light is safe. You do, and blue light you don't need to have a shield. Is with naked eye you can see that. Okay, only thing what you have to purchase is orange color goggle, and that's it. Yeah, so through orange color you will see very nice bands. So at the end, it's cheaper version, safer version, and the concentration is like this. How you should use cyber gold. So I have tried at least
Yes,
10, 15 different dyes, but this is one of the best. And this is good for the DNA, for RNA, and also other RNA species, for example, microRNA or so. Okay, is there any question?
And green master mix.
Can you please repeat the question?
No, I just said we are already using a GoTag master mix, that is a green master mix. So there also we don't need to put ETBR in the gel. But somewhere we have to, when we have to go send the samples for sequencing, then we cannot use this master mix.
Okay. Okay. I never use the colored one, the green one, master mixes. Yeah, you can load directly on the gel. I always like old school because whatever changes or these new tricks are coming, they are good, but you should not change your laboratory always according to the companies. You have to have one standard procedure, uh, and then you should have one or two people working only to test what is coming new. I am not against new things, but the thing is that these new things like the color buffer or color master mix, everything is good. The problem is they are not compatible. Cost is there, so they are not compatible with downstream analysis, although they say that they are compatible because they have to sell. But problem is the quality goes down of the downstream analysis. So either you have to go for the cloning or sequencing or other thing, the quality of that goes down. So I recommend you to purchase normal Taq polymerase and a DNTP. Use 6x dye. You can make your own dye. Why to purchase? Okay. You can have xylenol and glycerol or polyethylene glycol, water and TA buffer, and that is your loading die. So loading die is not something amazing. It has only 4 components. You can make your own dye in milliliters and use it. Okay, so I don't think there is need to spend money in this fancy stuff. You can save that money and go for NGS or something other good things. Is ready made, yeah. Is ready made. And cyber gold is one of the most sensitive in the market. Even it is sensitive than ethidium bromide. Okay. The next one is Eva Green, which is also sensitive, is as sensitive as cyber gold, but Eva Green's peak is at 500 because it's green. It should be 500. I need some in red range, not in green range. So cyber gold is good. किती वाजले रे? 1? Okay. So when is lunch? If yeah. Okay. So if there are LAMP PCR LAMP LAMP PCR is quite different story actually. Somebody is using LAMP. No. So the LAMP is quite different story. It is not part of our presentations. If somebody is interested, yes. But otherwise, I don't want to spend time on LAMP. Rajpal, are there any questions? If no, then we can stop. If you have any questions, then ask me. If no, then we can close the session.
Okay, today's session is over.
I'm winding up this session because I'm told that we are going today to ICMR or yesterday, and then we are going to Hafkin.
I'm sorry, sir. May I ask one question?
Yes.
Yes, last question, sir. Sir, actually, when you run the
can you write the question?
On Sanger.
Please write the question.
Sir, what is the protocol?
Yogesh sir,
how to
please write your
state
question.
the
You are not audible properly, Yogesh sir.
Yeah, I had already written, but actually it has missed.
Where is it?
Yes, I'm writing. Yes, I'm writing. Okay, I'm writing.
Okay. We'll wait for that. So actually, I was told to wind up a little bit earlier this part because we are going to this yesterday's and then from that we are going to Hafkin. So yes, yes, you can use. Yeah, yeah, yes, yes, you can use. No problem. How to set a program for Sanger sequencing? How to set a program? Program, I mean thermocycler program, right? Is it thermocycler program?
Yogesh sir, can you audible? Yogesh Chatur sir.
Okay, the Sanger sequencing on a PCR is standard. The Sanger sequencing protocol is the same for any gene. So whatever we set the protocol according to the
No, sir. When we run the uh,
tell him to write.
sequencing, that is Sanger sequencer,
Please write in the chat box, Yogesh sir.
3750. Yeah, 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 yeah.
Yogesh
No, no,
sir, please write in the
no
chat box.
thermocycler.
Yeah. So steps involved in the Sanger do you want to do the Sanger sequencing in your laboratory? You can do, no problem. It doesn't need anything actually, and it is extremely cheap. The Sanger kit, big die terminator kit comes with big die 3.1 terminator. So it is called as big die Sanger sequencing chemistry 3.1, and all the Sanger sequencing machines are compatible with 3.1. Do not buy 1.1. It's older chemistry. 3.1. Big die terminator. Big die terminator. Right? So let's go for that. Okay, so what you need for to set up the Sanger sequencing chemistry in your laboratory and purify that and send it to the company to sequence. So do you remember yesterday that lady told us or told you that if you make that, she will give you in 1 day or in 1 or 2 hours, 2 hours. But the results will be in 1 or 2 days, in the same day actually. So what she will do, she will put 10 microliter of water and put in the machine. That's it. Okay. Anybody knows how it works when it goes to company? No, nobody knows. So it's very simple. They take 6. Okay, so 6 nanogram of PCR product. So if you send them 100 nanograms, they have to dilute. Okay, so maximum they can take 6 nanogram of PCR per microliter should be the concentration, and they take 2 microliter, that is 12 nanogram total. So it comes PCR product 12 nanogram. Okay, now they add big die terminator 2 microliter, buffer which comes with big die 2 microliter. Okay, let's talk about primer. Okay, so primer usually you use is 10 picomole, right? You have 100, it comes 100 micromoles. Let's, yeah, you make the stock solution of 100 micromole. Your primer is 100 micromole or no? How it comes? They tell you that the company tells you how many microliters of water you have to add, right? And you add and you dilute that 10 times, and that is your working solution, right? Now you send this working solution, and usually what we do is we dilute this 3 times again for the Sanger sequencing. So it comes to 3 3 micromole. So this is 10 micromole, 10 micromole, 3 micromole. And this 3 micromole goes, so this is primer, only one primer, not forward reverse, only forward, for example. So 3 micromole is 1 microliter. And let's say we add 2 microliters of DNA, okay? 2 microliters of DNA, 4, 5, 6, 7, 3 microliters of water. That's it. So this is Sanger chemistry. This dye includes DNTP, DDNTP, polymerase, everything. You don't have to add anything. Okay, so cost of this is 1,000 euro per 1,000 samples. 1 euro. 100 rupees. But we have developed the buffer in our laboratory. This is trick. We never tell to company, but we dilute this 6 times and then use. Okay, so at the end cost is 6 times less. Okay, and well, here is total is 10 microliter. Okay, and this 10 microliters you have to put in the thermocycler, and cycling conditions are may vary a little bit. So 95 degrees Celsius for 2 minutes, 95 degrees Celsius for 30 seconds, 50 degrees Celsius for 30 seconds, 72 degrees Celsius for 30 seconds to 1 minute up. But I have put 1.30 minutes. Okay. 50 degrees Celsius is your annealing. It is standard for all the primers. Okay. Now your PCR looks like this. No, there is no. You don't need the final extension. Ah, yeah, no. Sorry, 35 cycles. 35 to up to 50 cycles. 1.30 minutes. 1.30 minutes. So you need 35 cycles. Only thing that differs between classical PCR and Sanger PCR is the classical PCR looks like this. Like this, right? Is 95, 50, 72. The Sanger PCR looks like this. So this is different. Okay, do you know what does it call? A ramping. Okay, so the ramping time should be extended. Okay, so You have to standardize the ramping time for your PCR machine. But usually that ramping time is between 10 to 20 seconds. So it is slow process. It's not fast. It is slow process. So because as you increase the ramping, the length of the whole PCR will be extended. Okay. Sorry. Ramping is there. Yeah. All ramping is very slow. Okay. So you have to set your cycler for slower ramping. Okay. So but most of the modern thermocyclers, they you can change the ramping time and it is fine. So this is the main. And once you have finished your PCR, you need to purify the PCR. Okay. So there are two again techniques for the purification of this terminator reaction. Okay. We call it terminator reaction. First one is very fast. That means you need to remove your primers because you added your primer, right? You need to remove your primer. You need to remove all this garbage. So some people say don't remove, keep it there, but digest them. Okay. So there is one. Anybody uses that? No. So you have to add one chemical in that. So it is commercially available. I will check today. I never use. I am against that actually. So I never use. So you just put 1 microliter of that. You incubate 10 minutes and send it for the sequencing. Okay. Or you go for ethanol precipitation. Okay. And that ethanol precipitation is again old school technique. You precipitate your DNA with ethanol and send it to the company. But several companies accept PCR product directly from this cycling. You don't need to purify because they add maybe 1 microliter of that magic chemical and they sequence. Okay. And the cost come down. We apply €5 per sample for whole procedure. That means we receive the sample, we do all Sanger sequencing, electrophoresis, and send €5. If somebody sends the sample to me after sequencing, we apply only €1. So €4 is manipulation charges. Okay. Okay.
Sir, Yogesh, sir, question is that what are the parameters set when putting sample in Sanger sequencing? Last question.
Parameters set when putting sample in Sanger sequencer. Instrument, but whether he has Sanger sequencer.
Yes, we used to do in the Anand. Actually, when we were in the Anand, we used to do this. But how that parameters are set in the program, that I don't know.
Parameter in the Sanger sequencer. That is why 2 to 3 hours I have to give lecture of sequencer parameter.
Oh, sir.
Yeah, that's parameter I can tell you. It has temperature, voltage, current. All you have to set, the temperature of the inside furnace, we say. Then the time of the electrophoresis, pre-voltage, post-voltage. Yeah, there are several parameters for the sequencer. Okay, that is Avant. I'm talking about Avant 3100 or 3300. I do not know parameters for the new sequencing machines because they are advanced. but I am talking about classical sequencing machines like what